All right. Welcome everyone to another journal club. I hope everyone's doing well. So today we have the pleasure of having Finley Grover-Thomas present his results on climate-associated natural selection in the human mitochondrial genome. So it's currently under review in MBE, but I hope many of you could have read a little bit about it on bioRxiv. And Finley did a project with me a few years back for his integrated masters. He did his undergrad and integrated masters at University College London. He worked on the Ouroboros, so fear not. Although there's no O2K measurements here, he's an expert. So we published a paper together on mitonuclear interactions in larvae and the effect on mitochondrial function a couple of years back. And now he's doing his PhD with Flo, also in my department, and collaborating with other people from the department that are part of the UCL Genetics Institute. So yeah, he's going to tell us all about it. So the floor is yours.
Hello, everyone. I'm Finley. Thank you for the introduction, Enrique. So I'm a PhD student working with Flo. This project was all kind of born about of while we were waiting for other data for the PhD, we were looking for something else to do, and we kind of developed into this. So it's a bit genetics-y, but I'm going to try and skip over that and focus more on the interpretation-y side, which I think you guys will prefer and get more feedback from, and that'd be useful. So thank you to Lucy, Francois, Ida, and Flo. Lots of supervisors, lots of PIs, all very helpful. Okay. Why can't I? Okay. So this is the overview. I'll talk briefly about previous work and hopefully why you find this interesting. I'll do a very brief overview of methods because I think it's beneficial, just so you kind of know what I'm talking about. I'm going to quickly run through the two types of analysis that we did in our results, and then I'll try and focus a little bit more on these candidate variants, which is possible functional effects. Okay, so background, we're all mitochondrial people, but the human mitochondrial genome, as you know, it's very highly conserved, has 37 loci, including 22 RNAs, 13 protein coding genes, 2 ribosomal RNAs. It's variable in copy number. It's non-recombining, and it has uniparental inheritance. I'm aware you guys probably all know this, but just to get the facts straight. But another thing to consider, it has a very high mutation rate. And the reason this is interesting is because we can see the same mutation appear at multiple points in the tree. And we can basically have separate examples of the same mutation occurring, and we can look at what happens in each of these occurrences of a mutation. So we'll come back to this later, but this is kind of a key point to say that we have some ability to look at what happens to mutation in different scenarios. At this point, I normally go into a bit of a spiel about what mitochondria do and why they're important. So I tend to present this to more genetics-y audiences, but I feel like that's not really useful here. But to summarize, they do all these key cellular processes, the site of the electron transport system. They have all these OXPHOS, ATP synthesis, all these cool processes. And all of this is to say that mitochondria are really important in modulating how we interact with the environment. So we have examples from lots of different species and lots of different environmental stresses. Here's just a snapshot of a few. But these are really important studies to show that mitochondria do have an impact, and I feel like I'm talking to a friendly audience here. But we have pretty strong evidence to show that mitochondria do modulate in response to these different pressures. So importantly, lots of this experiment is predominantly either phenotypic assays or looking at the haplotype level. So this is an example from Flo, where we're looking at these flies in Australia. And if you look at heat tolerance, the A haplotype, which is in red, has high heat tolerance. The blue haplotype has lower heat tolerance. But if you look at cold tolerance, it swaps around. And another way this manifests itself is that you can see a decline in mitochondrial gradients and mitochondrial haplotypes across different latitudes. So you can see at low latitudes, where it's hotter, you have a higher frequency of the A haplotype, and so on. So all of this is to say that we have these declines across latitudinal ranges that in some way reflect adaptation to different environments. We see this across species. So this is another example from anchovies. So you can see as you get further north, we have increasing frequency of this clade B. And similarly, if you go further south away from the equator, you also see an increase in frequency of this clade B. This has been shown in lots of other animals, so killer whales, orcas, as well as different types of salmon, Atlantic and Pacific. But what about humans specifically? Well, one reason humans are interesting, at least if you take off an anthropomorphic hat, is that modern Homo sapiens inhabit an enormous range of different environments. So this goes from hot, arid, dry, very little water, to dense rainforests with high humidity, high pathogen load, all the way up to this on the right-hand side, all these really cold, can be cold and wet, could be cold and dry, different temperatures. So we've been exposed to all these different environments, and we've, in theory, adapted to each of them. If I look a little bit more closely at this kind of cold and wet scenario, we have phenotypic evidence that there's increased basal metabolic rate in indigenous populations that live in circumpolar regions. And we also have evidence that mitochondrial copy number, mitochondrial DNA copy number, increases in northern Chinese populations that live in colder environments. So these are important in showing that in humans, mitochondria likely have a role, but these tend to focus on phenotypes rather than specific genetic SNPs, genomic SNPs. At this point, I have to acknowledge that I'm not the first person to think about this. There's been a whole range of studies. These tend to look at the haplogroup level, and they didn't always agree. So the largest of these studies, which is this one over here, had about 1,000 full mitochondrial genomes, but they predominantly looked at the hypervariable regions. So we actually don't have a singular study or a study that focuses across the whole mitochondrial genome. These studies were all published in, well, between 2003 and 2009. And since then, we have a lot more genomic data and we have a lot more environmental data. So we're in a much better position to kind of go back and answer this question. So I'm going to very quickly go through the data sets I use, and then we'll get to the results. So the genomic data set we use is all publicly available sequences from NCBI. So I downloaded every single human mitochondrial DNA sequence off NCBI that I could get. There's about 65,000 of them, which is quite a lot. But then in order to do this, we needed to have location data. So we removed all of the ones that had no known location. So that took us down to about 25,000. For some countries, so predominantly the UK, Russia, Spain, the US, we have well over 1,000, often over 2,000 sequences. So we downsample those to 1,000, so they're not biasing the data set. And for some regions, we only have between 1 and 20 sequences, which is not really enough to make an accurate kind of assessment for that country. So we remove all those different countries. So at the end of that, we're left with 19,570 full mitochondrial genomes covering 80 countries. And one of the criticisms and one of the limitations of this is that we only have country level data. And this is because if we go down to the regional data, we go down to a couple of thousand sequences. And we're even less sure that an individual has been in a region for a while versus been in a country for a significant amount of time. There are two types of environmental data sets we use. One of them is latitude and modern annual precipitation. This is Bile 12, MODCLIM, for those who've used it. So this is just an assessment from current climate data of annual precipitation. And latitude is used as a proxy for temperature. So those previous studies I mentioned tend to use latitude only as a temperature proxy. So we've kept that one in because it correlates well with different temperature variables. But we've also wanted to include annual precipitation because actually maybe humidity and water availability is actually an important environmental variable to consider. This is the first set of data. The second environmental data set we use is from this package called PassClim, which is from Andrea Malika's lab, which has a set of very easy to access reconstructed paleoclimate variables from, depending on the data set, up to the last 20,000 years, 22,000 years. And what we do is we, per country, average the values over those last 20,000 years, and then all of the countries who say come from the UK or Canada would get assigned the same estimate for that country. These are, we can circle back to these later, but these are the environmental variables we use. So these are the reconstructed paleoclimate variables. The first, well, half really, are different measures of temperature. And the reason we use these ones is because they're meant to be more ecologically relevant. So rather than just saying mean annual temperature, you're looking at the mean temperature of the wettest quarter or the mean temperature of the driest quarter. So it just gives you a little bit more of kind of insight into the seasonality and ecological insight. The last few are to do with annual precipitation or precipitation more generally, which again is giving us a slightly different viewpoint. And this last one we do is leaf area index, which is basically a measure of the plant biomass in a given region. And we're using this as a proxy for kind of diet, realistically. So what are the key questions? The key questions here that we want to answer are, the first one is, has the mitochondrial genome contributed to local adaptation in humans? We're using climate as the selective pressure. The second question we're asking is it a single environmental variable that has driven this adaptation, or is it likely in response to different conditions? And thirdly, we have the full genomes. Are any variants that we detect localized to specific regions of the mitochondrial genome or OXPHOS genes and so on? Normally when I present this, I focus on the first two because I tend to find that people need lots of convincing that mitochondria aren't just purely neutral. I'm hoping that won't be the case today, so I'm going to focus a little bit more on part 3. Okay, that's the first bit done. So now onto the modeling bit, which, apologies, there is some equations. Myself and my brother share the same mitochondrial DNA. This is not necessarily due to the environment, but because we are highly related. So our null model is that any genotype you see is purely predicted by what we call shared ancestry, so how related two individuals are. This is what we'd expect if the mitochondria is evolving purely neutrally. So it is only, genotypes are only explained by relatedness. Our alternative model to start with is we have our model with shared ancestry and we add in latitude and annual precipitation. And these are our climate proxies to start off with. And what we do is we ask is, does this alternative model with latitude and annual precipitation better explain the pattern of a genotype's distribution? So can we better explain or better predict what genotype an individual has when we take into account a measure of climate? So this here is slightly adapted from what you might have seen in the preprint in response to reviewer comments, but basically we have the mitochondrial genome kind of unraveled along the x-axis, and on the y-axis we have a kind of a comparison between this alternative model and the null model. So we're asking, does the alternative model better explain the null model? The first thing to note is that for most sites, the null model is not significantly outperformed. So this is consistent with neutral evolution, which is not entirely unexpected. But it's important to note at this stage that our measures of shared ancestry was a significant predictor for every SNP. So this is important because it suggests that actually we are capturing the variance, we are explaining the variance correctly with our measure of shared ancestry, and this is consistent with the neutral theory. The key point I want to state here is that we do detect candidate variants. And when I say candidate variants, I mean these are sites where the alternative model significantly better explains the pattern of genotypes. Because one of the, again, limitations of working with mitochondria is that, in effect, you could argue it's a single locus. It could just be that everything is in strong linkage with each other. But to control for this, what we do is we say, if there are two SNPs that are both better explained by the alternative model, if they are correlated by more than 0.2, we purely take the most significant candidate. We ignore the other ones. So in effect, the candidate variants we have are somewhat independent from each other. All our candidate variants are recurrent mutations. They occur in multiple separate branches of the tree. And this is partly a consequence of our method. So it's much easier to detect an association when you have the same mutation occurring multiple times and responding to the environment in similar ways. If you just have a single mutation that occurs once, most of its variation will be explained by shared ancestry because it's all in one clonal lineage. Therefore, it's much harder to pick out an association with climate. That's point number one. I'm not going to labor it anymore. The second type of analysis we did is we replaced latitude and annual precipitation with one of these reconstructed paleoclimate variables. And we do exactly the same thing, and we say, does each of the alternative models better explain the distribution than our one? Because we have lots of different bioclimatic variables that we model for every single SNP, if you look on the left here, you can have some sites where multiple different models all perform better than the none. In this instance, we just take the most significant predictor as our assigned bioclimatic predictor. And similarly, if there's two SNPs that are correlated, we just take the one with the strongest overall signal. The reason we're doing this is to try and assess whether mitochondria are moderating local adaptation to different variables. If they're not, and the main driver is a single variable, we would expect the same bioclimatic predictor to be significant across all our different candidate sites. This is not what we see. So this is a slightly tricky plot to interpret, but this is just a measure of how many times each of our paleoclimate variables is assigned as the most significant bioclimatic predictor. And what you can see is that for some sites, temperature is very important, specifically the temperature of the warmest quarter, so quarter. But for other sites, actually the strongest predictor is our leaf area index, so the plant biomass or the precipitation. So our interpretation of this, and perhaps we can have a discussion about this, is that this is consistent with adaptive responses to subtly different environmental pressures. And I also wanted to raise at this point that I feel like there's a lot of focus in the field on temperature, and we actually see that precipitation and precipitation components are also really important. We're done with the models. Now on to the candidate effects. So this is another plot from the paper, you see. So on the x-axis, we have position, which is useful just because we need to refer to specific sites. We talk about them. And on the y-axis, we have the mitochondrial locus. The color of the tile is the mitomap classification for that locus. So for example, this orange one is the origin of H-strand replication, and it's designed as origin of H-strand replication. So what do we see? Well, the first thing to note is that we have 11 of our 18 candidates are within protein coding genes. Of these, the 4 strongest candidates are all within complex 1. And I don't think this is potentially a surprise to any of you. It's very consistent with what we see from phenotypic studies, but it's nice to have a kind of genomic signal reflecting the same thing. Of the complex 1 subunits, we have ND1, ND2, and ND4 being the strongest hits. And this is consistent with what other genomic studies have found looking at hares and mountain monkeys. So it's nice that we have some kind of consistency across species. At this point, I'd like to say interpretation is not my strong suit. If anyone has any ideas, that would be beneficial. Our initial idea was about ROS flux and thermogenesis. So if you're at colder environments, actually maybe having higher ROS flux is beneficial. It generates more heat. But one of the reviewers very much didn't like that and suggested that actually it's probably to do with membrane fluidity, which you think is also a good idea. So perhaps this would be a good point to have a little discussion on later. For those of you who are quick at maths, you'll realize that there are 7 candidates in non-protein coding regions. We have 2 within tRNAs. We've got tRNA glycine and tRNA threonine. The tRNA glycine candidate is in the variable loop, and for the threonine is in the anticodon loop. We also have hits in both RNA subunits and throughout the control region. It's worth noting that we have lots of hits within the control region, but these are the hits that tend to be very highly correlated with each other. So we actually only have 3 separate candidates that are what we would define as independent. And very quickly, it kind of, so I get towards the end, I'm just going to focus on these 2 SNPs, which I think are interesting. The first of these is position M250, and this is slap bang in the mitochondrial DNA transcription factor 1 binding site. It is the single strongest candidate in our latitude and precipitation analysis. And we see that it's at the derived value is at highest frequencies in cold and wet environments. And the reason I think this is interesting is because, as I mentioned earlier, we have phenotypic evidence to suggest that in indigenous populations that live in circumpolar regions, they have a high metabolic rate. And in those Chinese populations, they have a high mitochondrial DNA copy number. So here we have evidence to suggest that potentially that is being mediated by the mitochondrial genome itself rather than a nuclear moderator. So I think this is a nice little kind of example that ties in with previous work. The second one, SNP that I would like to talk about is this position in the 16S ribosomal subunit. Also one of the strongest candidates we get, also at highest frequency in cold and wet environments. So on this axis here, you have, on the left, you have places that are very cold when they're very wet. And you can see here that it reaches a frequency of up to 30 or 35% in some populations. So it's reaching pretty high frequencies in certain regions. We know this is actually the plot I showed you earlier, is occurred at least 4 times. But the interesting thing about this one is that, according to other molecular biologists, this is not work I've done, but it has severe structural effects. And it's been associated with a number of different pathologies, including cyclic vomiting syndrome, migraines, glioblastomas, none of which sound very nice, but it's particularly interesting given that we see this frequency at 30% in some populations. So one possible explanation of this is that in colder environments, the SNP is beneficial, but when you're not in that environment, it has these maladaptive effects. Slightly complicating the picture on this one is the fact that this SNP also overlaps with a different locus, which is the short humanin-like peptide 6. So this is an peptide encoded by an alternate reading frame. There's quite a few of them in the mitochondrial genome. And of all the short human-like peptides in humans, this is the, or not just in humans, but mammals, this is the only one of these peptides that has really highly conserved start and stop sequences. And this is important because it suggests it has quite an important functionality. Also worked by other people, but it's been associated with retrograde signaling for thermoregulation, which matches what we see in terms of higher frequencies in cold and wet places. So again, interpretation is a bit vague for this one, but is there an apoptosis role? Is it ROS inhibition? We don't really know what's going on. Again, thoughts welcome. So with that, I'll just wrap up. I've gone through this quite quickly, but in summary, we have evidence to support the hypothesis that mitochondrial DNA has contributed to local adaptation in humans. And I don't think this point by itself is a trivial one. It's been debated quite a lot. I think it's nice to have some more evidence to support this idea. There's no single obvious environmental pressure. Some appear to be more closely linked to precipitation, some more to climate, some to biomass. So this could be responding to diet, thermoregulation, all this sort of stuff. So we don't really know. And finally, complex 1, the ribosomal RNA, and the transcription factor binding sites are loci of key interest. With that, I'd say thank you very much to Flo, thank you to Ida, all the different lab groups, to Francois and Lucy, and all of those who have published mitochondrial DNA, uploaded to NCBI, specifically those who've included location data. So thank you. And before I take questions, I'd like to quickly do some promotion for that. This is something I've been working kind of on the side with Enrique and Bella. And this is an R package designed to work with Ouroboros data, or not just Ouroboros data, but kind of mostly flies, but could be adapted. Work with mitochondrial biology kind of measures. We're in the early steps, but I could do with some people, if they're interested, just a couple of people to test it out, see what works, see what doesn't work. And if anyone's willing to help develop it, then the more the merrier. So thank you very much.
Thanks, Finley. It's great. I hope there's people that want to try the package as well. I guess people in the Ouroboros at the company have maybe their own R packages. I don't know. I think that's something we discussed in the past, but also with the new data lab, I think they're like integrating more the analysis. So maybe there's also something in development on their side, but it would be good if people could test and refine your package.
Yeah, I mean, I personally would love to also check it out and, be a bit involved there, because I think, yeah, we had some interesting conversations about, integrating, R and Python and various, scripts and stuff to make analysis far easier. So I'd definitely be interested in being involved. So I might reach out.
Yeah, that'd be good.
Great. Yeah, I have questions, but I'll let other people step in first. Does anyone have any questions, insights, explanations? What could be happening there with ND1, ND2, ND4, was it? I mean, I just stuck on the complex ones. So
Yeah, ND1, ND2, ND4. ND4 has a lot of hits in particular.
Neil, yeah.
Hi, sorry, I'm just adjusting my light level so that's a bit better. Yeah, thanks so much. Really interesting talk. I wondered whether you could, I mean, you probably don't want to know this, but whether you could do something more with your bioclimatic variables, because if I understand right, you've kind of considered them individually, whereas in fact they probably interact. Because I'm just thinking about the fact that productivity is greatest where there's both warm temperatures and precipitation. Just having the Sahara Desert doesn't do anything for you, nor does having just lots of precipitation. And in fact, having lots of precipitation and cold is actually very challenging to humans. I can speak from experience living in Glasgow. So I'm just wondering whether you could use a compound variable that actually captures that, where the most challenging environment is either cold and wet or warm and dry.
Yeah, so this was one of the big problems we had, because as you say, they are all indeed correlated with each other. And one of the ways we were thinking of doing this is using a PCA and getting a kind of, as you say, a compound variable. But the problem with that is that if you have a significant hit, then you still have to untangle which factor is contributing most to the principal component that's important. So with the first models we use with latitude and annual precipitation, we do a test where we add in the environmental principal components. And we see, are we, with our latitude and annual precipitation, are we capturing the environmental data, or is there lots of stuff that we're missing? So we try and see that, and we don't see any improvement in our model. There's no difference. So which would imply that latitude and annual precipitation are capturing kind of the breadth. As for working out the relative importance, this is one of the things that we really struggled with. I think one of the things we like about this approach is that it's, even though you're comparing the same thing repeatedly, it does give you a sense of which variables are consistently the most important. So as you said, cold and wet is important, and these are the ones that we see are the strongest predictors for our strongest variance. So both the MT250 and the M503010, the strongest predictor is how the temperature when it's cold and wet. But yeah, it's a good point. It's one of the things that there's all sorts of complicated analysis you can do to work out kind of the relative importance of correlated variables, but it gets very complicated very fast, and it's not necessarily that intuitive to interpret the results, even if you do get them. Yeah.
Yes, Jim.
Hi, thanks. I don't think my camera's working, but that's okay. Oh, here we go.
Yeah, you just appeared in a field.
I'm going to maybe rock the boat a bit.
Go for it.
Now, the data you get is country level.
Mhm.
Is there any way to control for geographic origin, which is a fancy way of saying race? If you get country data from the United States, for example, many of those people originated and presumably evolved in West Africa. So how do you marry that with the climate data from Alaska or Minnesota?
Yeah. So this is a good point. There's a couple of things we do. The first of which is that the data from most studies, when someone is classified as being from a country, it's they have 4 grandparents from that country, which is some level of control. And the other thing, as you say, about in America, you have lots, well, not just the USA, but throughout the American continent, you have large levels of admixture. So what we do is we completely remove all individuals from the Americas, from New Zealand, from Australia, and we rerun the analysis to see if signals there are biasing anything, and it doesn't change anything at all. The only thing that changes is that 2 SNPs are no longer significant, and these are SNPs that are common in Latin America. So you lose the derived variant. So yeah, we've done our best to account for that. Okay.
Any other questions? I can step in maybe with the, so I stuck on ND4 with the interpretation. I mean, I know, so some of your comments from your reviewers were pointing towards membrane composition maybe playing a role there and affecting the fluidity, and so the activity of complex 1, I guess. I mean, I'm making a shortcut, but yeah, something along those lines they were saying. But the ND4, I remember the recent studies by Sophie Breton's lab. I don't know if you saw them with the open reading frame encoding for some small peptides inside ND4. And those peptides themselves controlling, well, actually, or regulating to some extent by mitochondrial function and gene expression. Could something like that be happening there, you think?
Yeah, so I had a look at this actually for some of them. So the most significant hit, as far as I can tell, doesn't overlap with any of the ones that they list, the alternative
Okay.
important ones. I haven't checked all of them actually, to be honest. But one of the weird things we get is that, if I go back to this plot here. So what you can see here is that these represent the amino acid substitutions. And for lots of our significant hits, they're synonymous. So one of the explanations we have for this, or routes of avenues to look down, is that they are indeed involved in these kind of alternate open reading frames. So this is one of the ideas we had. It's also worth noting that in our fly lines, we have flies that are only separated by synonymous SNPs, and yet they have phenotypic differences. So clearly something is happening there. We just don't quite know what, I guess.
Yeah. And also, so how does this link to your, maybe you want to talk about the rest of your PhD, the other chapters you're doing, but like on your population, Drosophila population studies, are you going to be able to make a link between those variants maybe, and see if you find the same variants in the populations? Is that the idea? So how do you get from this to functional is what I mean.
I see. Right.
At some point.
I think so. For context, part of my PhD that I'm working on with Flow is that we have these experimentally admixed populations where they either have northern mitochondria or southern mitochondria from Australia, but they start off with 50-50 nuclear ancestry. And the idea is that over time, if you have northern mitochondria and you have a northern nuclear variant that's beneficial, that will increase in frequency, or the one from the same population as the mitochondria. So I think in an idealized world, and if everything goes perfectly, which rarely happens, what you would see is you would see the strongest signals in these sites. So these are the ones that are most important for function. I think it would be hard to go directly from here to phenotype without further work. You would need to do more stuff. I mean, it's possible that, I mean, can't remember where the mutations in our current lines are, but if we did some kind of analysis of the bar, the Cox, the lines that we have to date, and worked out what those mutations are, then maybe that could be a stepping stone, I think.
Yeah.
Yeah.
Yeah, I'm kind of surprised as well. There's nothing, well, maybe it's not surprising, I don't know, but I was thinking about UCPs, you know, what, like, you know, if those changes are somehow affecting coupling and heat generation and stuff like that, you know, linked to climate variation, why no UCPs, for example? Isn't that, you know, an easier target? So,
I'm not up to speed yet. Like, how would that? Because we just have the mitochondrial genes. Which of the?
yeah. Okay. Yeah. No.
Yeah, so
you're
we
right. Yeah.
just got mitochondrial genome. So ideally, we'd want to look at the nuclear genome as well. But there's no really good data set unless we go to all the biobanks in the world, which would be fun.
Right. Yeah. Okay.
Yeah. So the next step will be looking. So this just focuses on the mitochondrial genome. The next step is looking at the nuclear genome and the mitochondrial genome together to get a better idea of this interaction. But this was very much like a first. It was meant to be a side quest and kind of turned into a more prominent main quest. But yeah, this here is just mitochondria. There's no nuclear component.
Maybe I completely misunderstand what AlphaFold does, but can't you predict what the functional consequence would be with that?
Good idea.
Considering these mutations, yeah.
Yeah. That's a good idea. I haven't thought about that. So I think the changes in structure for the ribosome candidate, I think that was not from AlphaFold, but from a earlier equivalent program. So it would be a good idea to do that actually.
Anyone else has any questions or wants to feed some data to Finley for his tidy or uses tidyr script as well for their analysis? Don't hesitate to send him an email or just to me directly. But yeah, I think it's tough here for you know, you've got these associations, but what do they mean functionally?
Yeah, that's the next step, I guess.
Have you got any good suggestions from the reviewer as to the various reviewers as to what, you know, which direction this could go?
So I think most of the reviewers were focused on the like the genomic curation aspect. We had one reviewer who was talking about the kind of more physiological side. So he suggested that it was to do these candidates in ND2 and ND4. It's more likely to do with either membrane fluidity or like hydrophobicity, which could be something that's not really my area of expertise, but I think it could be an interesting one. I think part of the thing of this is that it was like a kind of a first step, and we want to try and look at the interactions more now. So it would be nice to see if we see the same genes showing up later on, rather than because it's very hard to do work on humans, right? Finish work on humans. So, and kind of the way that different traits manifest in humans is complicated. So complex disease. So it's kind of you're getting into a whole web there. But it would be cool to go into. Yeah.
And the last thought, ATP synthase, that ATP6, do you know which predictor was more strongly, or can you know which one was more strongly associated to this, to this loci being?
Yeah.
Okay, you've got, oh yeah, you've got a table, yeah.
Where are we?
Precipitation, the last one, precipitation of the driest quarter.
Yeah, and this was the one that I didn't
Response to cold.
as well. Yeah. Yeah. Yeah, that was one of the candidates that's already been identified using a different method. So it's nice that we get the same thing.
Yeah, that's very cool. I mean, I do agree with Jim's point earlier. I was thinking also, like, in terms of country specific stuff, it's a shame not to have the resolution that you could have. For example, when I see Russia, I imagine, Siberia and the rest, and Western Russia must be completely different.
Yeah, there are some countries, I mean, some of the ones that you would expect. So Russia, Canada, really variable. India is incredibly variable in terms of precipitation. So we did also run it without Russia, without the US, without these countries with really high variability. And again, the results don't change. So it's not like the results are being driven by that single country. But I think there's more, if you had better resolution data, there is a lot more you could do with this. But I think you are fundamentally limited by the quality of the genomic data curation that's out there, which is frustrating because this is publicly available data, and there's, as I said, there's 65,000 genomes that you could have used, but in most of these cases, location is just not included.
So you end up with a third of the database, basically. Yeah.
And even then, there's no, on NCBI, there are some fields that you can just kind of call very quickly, and location is not one of them. So often you have to go searching through the notes to try and find it and work out where it is from. So it's not very clear cut at all. Yeah.
Great. Does anyone have any other questions, thoughts, comments? If not, thank you so much, Finley. I think we can stop the recording here.